It's an honor to be participant to this symposium, actually. Tough act, to follow up on this very inspiring, molecular and technological talks. My work is very biologically, I would say, disease-oriented, medical-oriented. I've been studying now for almost, 3 decades, what I believe is to be an important, biological process, which is a vascularization, the development of blood vessels. I think it's very intuitive that your blood vessels are very important in physiology. It's very difficult to have any growth, anything meaningful in a multicellular organism unless you have your blood supply with the delivery of nutrients and oxygen. So the blood vessels are known to be important for millennia. What has been a little bit more recently appreciated, the blood vessels can play a role in some diseases, some very important role, actually. One of the diseases which was probably first associated with excessive blood vessel growth or angiogenesis is cancer, actually. I'm not going to talk much about that today. At about the same time, actually, the first studies on cancer were done in the late 30s. In the late 40s, came this really important, very influential paper by an ophthalmologist, Isaac Michelson. At the time, he was working in Glasgow, and then he moved into Israel, to continue his work, and largely based on very extremely insightful clinical and pathological observation, actually looking at the diabetic retinopathy. He concluded, that the disease retina, the ischemic retina, must be producing must elaborating some compound, some blood vessel growth stimulating factor, which later on actually was called factor X. For example, he observed that even though there is a disease in the retina, you can have a growth of blood vessels in the iris, which is in the front of eye. So it would imply that this molecule can be actually secreted by the retina, go across the vitreous, and then reaches the iris. It gives rise to this to a condition actually which is known as rubeosis of iris. And so there has been for many decades this search for this hypothetical molecule, because let's be clear, this very purely hypothetical molecule that that in cancer or in development or in die could induce angiogenesis. And actually, largely because of the work done by Judah Folkman in the early 70s, there was lots of interest in identifying this molecule. And actually, in the mid-90s, when I started becoming interested in this field, there was already quite a bit of a body of work. There was as many as a dozen of angiogenic factors which have been identified. Actually, mostly proteins, but not all proteins. And probably the most prominent of those was basic FGF, fibroblast growth factor, which is a molecule which is well known, for example, to neurobiologists, which is a very pleiotropic molecule. A molecule can do just about anything, And it is a very potent angiogenic factor. But something very, very puzzling emerged, in the mid 80s when the FGF was cloning. It does not have a signal peptide, so it's not a secretin protein. And so you can immediately appreciate it just looking at this slides, actually, that the angiogenesis almost, intuitively require the release of some molecule in the microenvironment, because it needs to be created some kind of gradient, some kind of biochemical gradient, which attract, blood vessels. So in other words, a molecule like FGF, even though it was pharmacologically very potent, but it didn't seem to be really so well suited, to regulate angiogenesis. Even experiment with antibody, even later on knockout, it didn't show that an FGF all these other molecules were important. And actually, in the 80s, actually, when I started my work, I was a postdoc actually across the bay at UCSF. Actually, I was working actually in a completely different field. Actually, I was working on endocrinology, on the pituitary. Actually, part of my postdoctoral project was I was able to isolate and characterize some cells known as follicular stellate cells, which are rather obscure cells in the pituitary, which is, however, seems to already not, to have some unique kind of, interaction with blood vessels. They ended up in close proximity with the blood vessels in the pituitary. So suggesting some kind of role in regulating this process. And actually, in the course of the years, actually, I observed that the cells release in activity. Just at that time, in the 80s, there was all these factors uncharacterized, etc. I thought it was very, very intriguing. Over the years, I was able to isolate, to purify the protein. Those were the days where to go to the DNA, you have first, to purify the protein, sequence sequence, and then based on the amino acid sequence, design probe, for cDNA cloning. And in 1989, actually, I joined Genentech at that time. We were able actually to clone, this molecule that we call vascular endothelial growth factor. And at that time, we didn't know whether it was an important molecule, but certainly with some interesting features. For example, it's a secretin molecule, which is kind of emphasized in this paper, unlike this other molecule. So potentially could be released into the microenvironment and play a role. But that's all we knew at the time, and we didn't know whether this was going to be just one more angiogenic factor. And actually, it took several years, but this is one of probably the most, still I believe, one of the most, compelling experiments which indicate, the VEGF was an important molecule. This is the VEGF, knockout in mice, actually. And you can see this is the yolk sac of the wild type. You see this is very nice. This is a beautiful vascular supply in the yolk sac. And this is the VEGF, mutant, which lacks only one of the two alleles. So there is still 50% of VEGF. And as you can see, there is hardly any blood islands in the yolk sac, and the embryo is already severely underdeveloped. It's probably already dead. So this seems to be an important molecule. Seems to be one of the very, very few examples, at least in the mammalian genome, of this kind of critical dependence on a factor for the vascularization. Actually, this haploinsufficiency. And since then, I think we learned a lot about this molecule, and now actually. It's a very well established, very well characterized, system. Actually, there is, some receptors, some tyrosine kinase, receptor. One is the best known is VEGF, receptor 2, as well as some co-receptor like a neuropilin, which is also, a molecule implicated, in neurobiology, So the activation by VEGF, this tyrosine kinase receptor, result in a signaling cascade, which is not that different from other tyrosine kinase, like the PDGF, receptor. And now we understand, VEGF is important in a variety of physiological processes, and even in the adult. It became a therapeutic target, actually. I'm not going to show this data in the interest of time. Already it became known that they block, VEGF, either the ligand or the signaling cascade can inhibit tumor growth. And that result actually in therapeutic application, therapeutic intervention. There is today as many as 12 different VEGF pathway inhibitor, which has been approved in therapy. It's been approved in cancer therapy, for example, in colon cancer, in renal cell cancer, as well as in a variety of malignancies. But admittedly, I think the most interesting, perhaps the most impactful application is not in cancer, but actually is in dye. Cancer is such a clever system, found a way, almost a diabolic way, always to evade and to develop resistance. Seems to be that this problem was less in dye. Let me first point out, actually, this slide is derived from cancer, but applies virtually any system. VEGF is induced by hypoxia, by now well-known and well-characterized mechanism. There is a transcription factor called IF1 alpha, as well as others, actually, which result in upregulation of a series of hypoxia-inducible genes, including VEGF. And so this is really is very important physiology, because whenever you have hypoxia, actually you need more blood vessels. A molecule like a VEGF is released, and actually create new blood vessels. And so the question is a VEGF is this hypothetical factor X at least related to this hypothetical factor X hypothesis postulated by Michelson in the 40s. Actually, once again, there's some interesting characteristic, like it's a diffusible molecule. Also very important, it can be induced by hypoxia, because hypoxia and vascular damage are kind of key features of this disease in the eye. And actually, just to illustrate you like how the eye at the retina in a patient with a diabetic retinopathy, which is one of the key key ischemic retinal disease looks like. You don't need to be an ophthalmologist to appreciate, this very extensive vascular disorganization of these vessels, which are really so abnormal, tortuous, and there is extensive bleeding, which can result, essentially in blindness, and actually this illustrates some of the characteristics, of these vessels. Actually, they lack, some key elements, in the vessel wall, which are called pericytes, and the loss of this pericyte result, in this very abnormal vascular bed, which is very leaky, and tends, to bleed, and so this result in this actually in some of these key events, And actually, almost over 20 years ago, actually, using at the time some very sensitive assay which was developed in our lab, in collaboration with a group at Harvard with George King, and colleagues, we're able to show that the levels of VEGF are elevated in the eye fluid of patients with ischemic retinal disease, like the diabetic retinopathy of prematurity, and also occlusion of a retinal vein, which is another disease resulting in ischemia. There was almost a perfect correlation between the levels of VEGF, in the eye fluid, and actually, angiogenesis. And this is actually an animal experiment, which at the time actually was seems to be very compelling. This is a primate model of, of laser-induced, occlusion of the retinal veins, which create, this ischemia and release, VEGF. And this is actually this intensive angiogenesis in the iris, just like, I was at the very beginning alluding to Michelson, idea. And as you can see, an antibody to VEGF almost completely actually inhibiting this process. At the time, the animal data were very compelling, but there was a lot of skepticism that this could have really any clinical relevance, because many people, it was not, that this event, happened in this animal model within 2 or 3 weeks, and then the blood vessels regress. Instead, in the humans, this disease, takes place in years, sometimes even in decades. So this was a little bit, kind of, slow down, the process, the decision to develop, a VEGF inhibitor. But there was actually another disease which was really has been historical and unmet, medical need, which is age-related macular degeneration, which is the leading cause of blindness in the population over 50. This is some of the features of this disease, actually. To make a long story short, the disease has been historically classified in 2 types: the dry form and the wet form. The dry form is characterized by this by this structure. Let me go by order. The dry form is characterized by the so-called drusen, which has a series of bumps in the retina, and unless they become too many, they don't really impair vision. Instead, the neovascular form, the wet form, even though accounts for only maybe 20% of the case of AMD, is the one which is responsible for about 80% of the case of blindness. And so this is clearly an enormous unmet medical need. A work done over the last 10 years has showed that there is some very clearly genetic factors, some genes mutated which predispose to AMD. And there is some which are more important than others, actually. But the one which is very clearly has been the most important, the most strongly associated with AMD, is a complement factor H, which is a protein we actually regulate complement. Inhibits, is like a dampening system for complement. So whenever you have this mutation in complement factor H, there is an excessive inactivation of a complement, which normally is role actually to help us to fight pathogens. But anyway, if it not properly inhibit and regulate, can give rise to tissue damage and inflammation. And this is what is believed to happen in AMD, actually. And actually, this mutation in complement factor H seems to be actually a risk factor for both the dry and the wet form. This is our patients with a different kind of this disease, actually, as opposed to see. This is a patient with a diabetic retinopathy. Even though there is large areas where there is actually in the peripheral retina, where there is this black spot in the peripheral retina, but central vision, which is very critical for reading, for writing, etc., is relatively preserved. Instead, in a patient with AMD, at least with the severe AMD, even though the peripheral retina, the peripheral vision is relatively spared, you can say this is really actually the central vision, which is the one which is so severely impaired. So even individuals which otherwise could be relatively healthy are unable to read, to write. So the quality of life is really very severely actually deteriorated. And so this is actually, as I was alluding at the beginning, when it was initially believed that there is a dry and a wet form. Now it's very clear that this kind of, is a spectrum of, of AMD, actually. Area of, dry and wet, can coexist in the same eye. And actually, this is the way, actually, the advanced, AMD associated with the dry AMD looks like. You have this areas of a geographic atrophy. This is pretty broad areas, actually, with a depigmentation, which kind of expand, and eventually deteriorate, vision very, very significantly. And this is really what is, once again, the most, destructive form, which is the neovascular form. Actually, you have, this growth of vessels which come from, the choroid, which is an extravascular bed, and they literally make the holes across, membrane, which is called, Bruch, membrane. And these vessels can be very catastrophic because they can bleed and they leak, it can destroy the photoreceptors. And so at the time, in the mid-90s, actually, when all this work, was actually the story of VEGF, kind of, was developing, actually, there was lots of debate as to what kind of disease you may want, to target. There is a much more compelling scientific rationale to target, like the diabetic retinopathy or the ischemic retinal disease, because, VEGF was so clearly associated with this disease. But at the time, on the other hand, there was some pretty effective, therapy for this form, which is laser photocoagulation. And instead, there was really nothing for age-related macular degeneration, actually. But, admittedly, the evidence of VEGF was implicated was very weak. There was only some immunohistochemical data showed that there is a VEGF expressed, in this choroidal neovascular membrane. But on the other hand, there was such an important disease, such an unmet, medical need that, it was decided, actually, to develop a clinical trial, actually. And actually, the difficult was what kind of molecule are you going to use, to do this, injection in the eye. No one has done any long-term administration of an antibody in the eye because it was believed would be much safer to inject, locally. So it was thought, an antibody fragment, which is an FAB, which is a monovalent antibody, which is devoid of FC, could be the safer form. And actually, this is actually the clinical data actually was presented, was first shown some time ago. Actually, at the time, this is a visual acuity with patients with an aggressive type of AMD, which is a classical form, which is extensive neovascularization. Patients actually were randomized, to receive, either this an anti-VEGF antibody, which is called Ranibizumab, which was injected, intravitreally every month, And actually, the green line is not a placebo; is what at that time was the standard of therapy, which is the photodynamic therapy. And as you can see, there is a little bit of increase, in visual acuity with the photodynamic therapy, and then there is a kind of unstoppable vision loss, And instead, with each one of these 2 doses of ranibizumab, but there was a very striking, at that time, this was totally unexpected. There is an increase in visual acuity. It was possible to show that about 35 to 40% of patients have this marked increase in visual acuity. In about 90%, you prevent severe visual loss. So this was all very unexpected. It is actually some of the concept, vascular biology actually show that by blocking VEGF, not only we almost completely inhibit further growth of this choroidal neovascular membrane, but the vessels are substantially remodel, and so this probably show why they leak less. But so the question is, what is the real impact, on blindness? Actually, the clinical data, the clinical trial data look very striking. There is this model, actually done, by a group at Johns Hopkins by Bressel et al. That predict, that if patients were treated, like in clinical trial, there was with the VEGF inhibitor, there would be a very substantial reduction in legal blindness. actually. But then the real world, data in the clinical trial data could be something very different, Usually, actually, the clinical trial, it's very difficult to fully replicate, in the real world, the clinical data, because a patient do not follow very regularly. But this is a very, very important study. It was published in Denmark, a study done in Denmark a few years ago. And look at the incidence of legal blindness, either due to macular degeneration, of this line, in black, and versus other forms of, of blindness, non-related, to AMD. And actually show that after the availability of VEGF inhibitor in 2006, there has been a steady, drop, in the incidence of legal blindness. Actually, there is about, 50% actually drop, So a similar data have been seen in other kinds. So I believe this is probably the most, striking result of, the angiogenesis field. I believe it very, very significant. And I would like to, I know that I'm running over time. I would like to make a point that actually there is a other approach. We have not really totally solved, the problem, because there is maybe 25 to 30% of patients which are not, effectively treated by VEGF inhibitor. And there is a geographic atrophy, which of course, is not a neovascular stage. Actually, could be where there is a clearly an unmet, medical need. And I was alluding at the beginning that complement seems to play a very important role in that. There is a number of strategies targeting the complement. And actually, even though there is no drug which has been approved, but there is some very preliminary data suggesting that if you inhibit, complement, in particular with antibody to factor D, you can at least, reduce, the growth of this, of the geographic atrophy. And as such, since I went over time, I will say that, this has been actually very exciting to see this start, from a basic research to some clinical data, but there is a much more to be done. And certainly, I've been very fortunate to have some incredible colleagues that over decades have been actually given a major contribution. Thank you very much for your attention.
A couple of questions. Maybe I'll just start. So now that Jennifer can knock out your VEGF receptor, what do you think of that? Or would you not risk lowering signaling by VEGF to 0?
Well, I think this is a case where actually the classical genetics, even the very powerful manipulation, but not terribly helpful. For example, I would like to say that you can never predict, that VEGF could be important, even measuring a panel of cytokines, etc., in the eye fluids, etc. But in terms of a knockout receptor, I don't think I would be very careful in doing that, because there is some evidence that actually even the knockout already was very illuminating. There is like a gene dose, haploinsufficiency. If you inhibit, the VEGF system very hard, you may actually promote, geographic atrophy. So actually, these days, people started backing off from this kind of monthly injection of, VEGF inhibitor. You have a PRN kind of, as needed, kind of treatment. In general, 4 to 5 injections per year is what you need actually to do that, which is obviously very, very fortunate.
Okay, well, thank you very much.